Hello, I'm Wyatt Emmerton from Franciscan University, and today I'll be talking about my research I've been doing with Dr. Perosic on G-coupled protein receptors. Now, G-coupled protein receptors have been extensively investigated for many years due to the important role they play in cellular signaling, and these proteins are characterized by their extracellular amino terminus, their 7 alpha-helical transmembrane domains, as well as their intracellular carboxy terminus. The objective of this research seeks to analyze the structural characteristics of G-protein coupled receptors and to essentially further uncover important residues which have been conserved across species. Additionally, we seek to investigate phylogenetic relationships between different receptors and essentially to uncover group-specific conservations unique to each receptor. The receptors which were investigated in this study were receptors for dopamine, epinephrine, sphingosine 1-phosphate, muscarinic acetylcholine, and neuropeptide Y, and serotonin, as well as kappa-type opioid receptors. In Figure 1 here, we can see the alignment of 1,100 different G-coupled protein receptors. This was generated using T-coffee and then manually adjusted using tertiary structures as guides. The conservation are as follows. Essentially, 6 columns were invariant. Those were in red. The green are 80 to 99% conserved, where we had 14 columns of those. And the blue columns represented 60 to 79% conservation, where we had 44 columns. The white are not conserved at all, or are below 60% conservation. Sorry. Here in Figure 2, we can see 2 different views of the invariantly conserved residues in the D2 dopamine receptor. There are 6 residues, and these play a role in hydrophobic packing as well as hydrogen bonding. Additionally, it's important to note that these residues lie in the core of the transmembrane helical bundle, closer to the cytoplasmic side. And the yellow structure in the middle is the ligand, which is bound to the receptor. In Figure 3 here, we can see we used conserve analysis to essentially map the conservation of the amino acids on a 3-dimensional model. So here we can see that the most variable amino acids lie on the outside of the transmembrane helices, while the most conserved residues lie on the inside of the alpha helical bundle. And then in Figure 4, we identified the 5 most well-conserved sequence motifs in the D2 dopamine receptor. Now, according to previous literature, amino acids from all 5 of these amino acids that were found in all 5 of these motifs contribute to the trigger region, while all the motifs except motif 2 contribute to both the linking core and coupling region of this protein. And moving on to Figure 5 here, we did a bootstrap phylogenetic analysis of the G-coupled protein receptor family, and in blue we see the D2 dopamine receptor, which is the one we've been focusing on here. We can see that the sphingosine 1-phosphate receptors are more closely related than the other groups, and the most distant is the muscarinic acetylcholine receptors. Now, to conclude, essentially, after going through everything, we aligned over 1,100 amino acid sequences, and we found that only 6 amino acid positions were fully conserved. Additionally, these conserved amino acids lie within the core of the alpha helical bundle. And then additionally, the most invariant residues actually face the lipid bilayer, which surround the alpha helical bundle. The five most well-conserved motifs also are the transmembrane alpha helices, all of which contribute to the trigger region, and all of these motifs except motif 2 comprise the linking core and coupling regions of the protein. Future research will involve the addition of more sequences from homologs to the current alignment, and then we will be reevaluating the conservation and phylogenetic relationships. And we will also be seeking to identify group-specific conservations which contribute to the unique function of each of these proteins. I think that concludes it. Thank you so much for listening, and I hope you enjoy the poster.